So hello again and welcome back for the most important and most interesting session of every year's DDL. And this is the Pat Brunella session. Today we're gonna find the Pat Brunella Award winner for the New Investigator Award. But before we let the finalists speak, I might give a little background of the award. So the Pat Brunella New Investigator Award had been created in the memory and recognition of Pat Brunella. She was an inhalation scientist, and she was in the committee starting off this meeting, starting off that conference, almost 35 years ago. And her clear vision was that it should be a conference where which is accessible, which is inclusive, and which actually brings those up on stage who actually do the work and do the science. If I may quote her, those who do the research should present their work, not their stuffy old boss or academic supervisors. So this is like the core of the conference, and this is why we today will let the young people speak. So, as I said, the award had been created in memory of Pat Brunella because she sadly passed away far too young. So this award really shall encourage young scientists from both academia and industry to submit, present, and also discuss their work here at the conference. So this year we had close to 30 submissions for this session, and we had a hard time to select 5 finalists, which are here today to present their work to you. They were selected based on the conference paper they submitted, and they will be judged based on what they present here on stage and based on how they will defend their work. So in that sense, we need your support. Please ask questions after their presentations so that they can actually show us how they know about what they were doing and how they can defend their work. For this session, we won't accept Slido questions. So really, please come up to the microphones and discuss and engage with our young scientists. So with this, I'll start the session, and Marie, who's our past year's winner, will introduce the first speaker. Thank you.
Yeah, I'm happy to introduce the first speaker, Brunella Grassiri. She is a final year PhD candidate at the University of Pisa, specializing in pharmaceutical and bioactive substances. Her research focuses on the development of drug delivery systems for ocular, pulmonary, and dermal applications, bringing together aspects of synthetic chemistry, nanocarriers, and biomaterials for biopharmaceutical purposes. Her work focuses on lung administration of off-label drugs and new antimicrobial therapies. So welcome, Brunella.
Thank you for your kind introduction. And first of all, I would really like to thank the organizing committee for giving me this great opportunity. For me, it's an honor to be here. So the project I'm presenting today is a deeply interdisciplinary project, starting off with pharmaceutical, medicinal chemistry, pharmaceutical technology and biopharmaceutics, microbiology, up to clinical microbiology. And myself, the one in the smiling picture above, had the chance to actively participate at each step of this research, also during my PhD period as a visiting student supported by an Erasmus grant. So starting off with some lung infection notions, Aspergillus fumigatus was included by the World Health Organization in the critical priority group of fungal pathogens that are associated with serious risks of mortality in humans. Moreover, pulmonary aspergillosis is particularly severe in patients with cystic fibrosis, in which it's clearly associated with high rate of mortality. So this WHO call, together with the rising rates of antimicrobial resistance, highlighted the need of new approaches to antimicrobial treatment. And an unconventional strategy could be to exploit microbial nutritional vulnerabilities. So in this aim, gallium siderophore compounds could be used. Gallium indeed shares many chemical and physical properties with iron, which is essential in microorganism metabolism. But under physiological condition, iron 3 plus is reduced to iron 2 plus, whereas gallium cannot be reduced. Siderophores, in the other hand, they are a small molecule that have high affinity to iron, and they are able to chelate iron and then to bind siderophore receptor, increasing the uptake of the metal. Interestingly, in the infection site, there is a greater demand of iron than in normal cells, and an upregulated ribonucleotide reductase that caused by rapidly multiplying cells. So in this context, gallium siderophore complexes with a Trojan horse mechanism of action could be exploited, in which the siderophores masks the metal gallium and increase the uptake of gallium into the fungal cells. And once in the fungal cells, the exchange of redox active iron with redox non-active gallium hinders the production of DNA and drives to apoptosis. So this could actually be a potential innovative approach for antifungal treatment. So our research aims to first develop a newly synthesized gallium siderophore-based antifungal agent against azole-resistant Aspergillus fumigatus, to then develop a formulation that will efficiently deliver to the lung this powerful drug, and finally to evaluate the efficacy of this selected compound in an in vitro lung infected model with fungal strains collected from clinical patients, So 3 different gallium siderophore compounds were synthesized, namely GAS1, 2, and 3, and these compounds were then tested and compared with 3 reference gallium salts already known for their antimicrobial activity: gallium nitrate, citrate, and maltolate. So GAS1 complexes resulted to be selectively toxic on Aspergillus fumigatus and non-toxic on human cells at their active concentration, and out of the 3 gallium siderophore compounds, GAS1 was selected as resulted fungicidal at a concentration of 0.5 mg per ml. So we had our lead compound. And then a formulation study was carried out both through a solution for nebulization, which was kept really simple, so a saline solution of GAS1, and then 3 different dry powders obtained through spray drying. These powders were obtained by spray drying a water solution of GAS1, adding different concentration of L-leucine, as detailed in the table, that was used as the only functional excipient. So the powders were then deeply characterized, and the geometric diameters of all the particles resulted to be suitable for inhalation, for pulmonary drug delivery. And they showed a nice corrugated surface, reducing the coadhesiveness of the powders and improving the powder flowability. And then all powders, based on their leucine content, showed a predominant amorphous behavior. So, yeah. And then the aerodynamic properties of the obtained formulation was determined with NGI, and we saw that all formulation actually showed good characteristics for pulmonary drug delivery. We saw an improvement in MMAD and FPF for the powders, and a trend of increased delivered dose and FPF with increasing content of L-leucine in the powders where L-leucine was used as a dispersion aid. Before moving to the next step of our study, the formulation were again tested again at DCC, Aspergillus fumigatus strain, and they resulted to maintain their activity against Aspergillus. So the calculated minimum inhibitory concentration resulted the same as the one of the unprocessed GAS1. So in view of applying a more complex in vitro model, GAS1 antifungal activity was verified in presence of artificial sputum medium, which contains mucin, iron chelator, DNA, salts, and amino acid. and which resembles the composition of mucus of a cystic fibrosis patient, and against clinical isolates collected at the University of Hospitals of Pisa from hospitalized patient. And once again, the activity of GAS1 was confirmed, maintaining the same minimum inhibitory concentration. So then we assessed an in vitro lung infection model. So basically, on translow insert, human lung distal epithelial cells were grown, differentiated on an air-liquid interface, and then infected with the clinical isolates collected at the University Hospital of Pisa of Aspergillus fumigatus. And then the samples, the insert was treated with GAS1. Two different experiment sets were carried out in this case. So we first stained the insert to see through micrographs the fungal growth. And then we investigated a dose-dependent effect, plating the samples on Saburo agar. What we saw, interestingly, was that the presence of human cells was able to boost the growth of Aspergillus. And so we wanted to see if GAS1 was active. And impressively, as we see from the pictures above, in the treated samples, no fungal growth is evidenced. And then a marked dose-dependent effect was also shown, when dose dependence from 3 to 6 logarithmic scale growth inhibition was evidenced. So in the end, GAS1 resulted to be active in presence on clinical isolates, in presence of human cells and of artificial sputum medium. So in conclusion, GAS1 resulted to be active against Aspergillus fumigatus, not just the ATCC strain, but also azole-resistant Aspergillus fumigatus, well tolerated in human lung epithelial cells, and formulated for optimal pulmonary drug delivery, both as a solution for nebulization and 3 different dry powder formulation. So an inhaled formulation that is able to efficiently deliver this siderophore gallium complex represents a promising tool for the treatment of Aspergillus fumigatus infection. So I would really like to thank my research group, especially my PhD supervisor, Professor Piras, Professor Anne-Marie Healy, and our research group, and to acknowledge our sponsors. And thank you so much for your attention.